Hello and welcome. Today, we're going to conduct a practical lesson in which we will learn to use a microscope. So far, we've covered some fundamental concepts about cells. We've learned that cells are incredibly small structures, so tiny that they can't be seen with the naked eye. To observe these cells, we need to use magnification tools, like microscopes. Now, we're going to bring all this knowledge into action with a hands-on practical. We'll be preparing our own microscope slides and using our microscopes to observe sections of plant and animal cells. This practical will not only help you see these cells up close, but also give you the opportunity to practice drawing and labeling cellular structures as we've discussed. Let's get started. Before we dive into the details of preparing microscope slides and using a microscope to observe, draw, and label sections of plant and animal cells, let's take a moment to consider why this practical is important and how it can benefit you. Observing cells under a microscope gives you a first-hand look at the fundamental units of life. It helps you appreciate the complexity and beauty of cellular structures in both plants and animals. By preparing and examining your own slides, you'll gain a more intuitive understanding of cell anatomy, which is important for grasping more advanced biological concepts. Preparing microscope slides and using a microscope are foundational skills in biological sciences. Mastering these techniques now will serve you well not only in higher education, but also in various scientific careers. This practical trains you in slide preparation, focusing on techniques such as staining and mounting, which are essential for clear observation. Additionally, honing your ability to use and fine-tune a microscope cultivates precision and attention to detail. This practical also emphasizes the importance of careful observation and accurate recording. By drawing and labeling the observed sections of plant and animal cells, you develop your skills in scientific illustration and data presentation. These skills are not only useful for exams, but are also critical for any scientific research or professional work involving data visualization and communication. Being able to accurately represent your observations helps you and others understand and interpret scientific data more effectively. On to the practical. Let's start by gathering our materials. Sam, what do we need?
So for this practical, we need a microscope, microscope slides, a pipette, an onion, a pair of tweezers, iodine solution, a lab book, and a sharp pencil for drawing.
Before we begin, let's quickly review the parts of a microscope. The eyepiece is the lens at the top that you look through, and it usually magnifies the image by 10. The arm supports the microscope and connects the eyepiece to the base. This section is used for carrying the microscope. The objective lens is located on the revolving nosepiece. These lenses further magnify the image, commonly by 4 times, 10 times, and 40 times. The stage is the flat platform where the slide is placed for viewing. This has metal clips, known as stage clips, that hold the slide securely in place on the stage. The condenser focuses light onto the slide from below, enhancing the clarity and contrast of the image. The mirror is just below the condenser and reflects light up through the condenser, stage, and lenses. There are also 2 focuses. The coarse focus is a large knob used for moving the stage up and down to bring the specimen into general focus. The fine focus is the smaller knob for making precise adjustments to sharpen the image. Now that we have reviewed the components of a microscope, let's go ahead and use one. In our first stage, we need to prepare the slide for the microscope. Start by placing a clean glass or plastic slide onto a flat surface and add a single drop of water to the center of the slide. This will help the specimen stick to the slide and prevent it from drying out. Next, carefully cut an onion and use tweezers to peel away a small piece of the outer epidermal tissue. This thin layer is ideal for viewing under the microscope. Still using the tweezers, place the onion tissue carefully onto the slide on top of the drop of water. Next, add a drop of iodine solution. The iodine acts as a stain, making the nucleus and cell walls more visible under the microscope. Finally, gently place a cover slip over the specimen. To avoid air bubbles, use a pair of tweezers to lower the cover slip at an angle, squeezing out any air as it is secured. This ensures a clearer view of the cells. In our next stage, we're going to use the microscope to see some cells. Secure your prepared slide onto the stage of the microscope using the stage clips. We need to line up our slide in the center of the stage, such that the onion sample is directly positioned for light to pass through. For now, set the objective lens to the lowest power, which provides the least magnification and a broad field of view. Begin focusing the image by turning the coarse adjustment knob to move the stage close to the objective lens. Then, look through the eyepiece and slowly adjust the coarse adjustment knob to bring the specimen into rough focus. Next, use the fine adjustment knob to sharpen the image until the cells appear clear and detailed. Now, make a record of your observations in the microscope. Use a sharp pencil to draw the cells you observe, ensuring your lines are clear and precise without any shading or colouring. At this stage, on the lowest power, draw the structure of the cells. Don't forget to title your drawing appropriately and mark the magnification at which you observed the specimen. After we have completed our drawing of the lowest power lens, we can return to the microscope and get a closer image of the cells. To do this, we need to change the objective lens to a higher power, such as 40 times. This will give us a closer image, but we need to use the fine adjustment knob again to focus the image. Once this is completed, we return to our observation stage to make a record of the image. This time, use a sharp pencil to draw a specific cell you observe, ensuring your lines are clear and precise without any shading or colouring. Accurately represent the proportion of subcellular structures within each cell, and label what you can see using straight lines that do not cross each other. Finally, title your drawing appropriately and mark the magnification at which you observed the specimen. Now, let's analyse our results. In this practical, there isn't a specific biological reaction occurring, but rather the goal is to observe and record the structure of cells. In today's practical, we specifically looked at onion cells, observing and drawing their structure and organelles. We can use these drawings to compare the structure of an onion cell with that of a typical animal cell. We can see from these comparisons the different organelles that plant and animal cells have. We also took numerous steps to enable observations of cells in this practical. Let's quickly recap them. Firstly, we added a drop of water to the slide as the water acts as a medium to keep the onion tissue hydrated and helps in adhering the tissue to the slide. This prevents the tissue from drying out and allows for a clearer observation. Secondly, we specifically removed epidermal tissue from the onion to study. The epidermal tissue is thin and relatively transparent, making it easier to observe individual cell structures under a light microscope. Using tweezers ensures precise handling and minimizes damage to the tissue. Next, we added iodine solution to stain the cell. This makes its features like cell walls and nuclei more distinct under the microscope. Staining significantly enhances the contrast between different cell structures. Furthermore, placing a cover slip over the specimen helps to flatten the tissue, ensuring a uniform thickness, which is crucial for clear viewing under the microscope. It also prevents the objective lens from coming into contact with the specimen and causing damage. Finally, we use multiple lenses to observe the cells. Starting with the lowest power objective lens allows for easier placement and initial focusing of the slide. Once a rough focus is achieved using the coarse adjustment knob, the fine adjustment knob helps to bring the image into sharp focus. A higher magnification can then be used for detailed observation. In today's lesson, we performed a practical experiment on how to prepare a microscope slide and use a microscope to observe, draw, and label sections of a plant cell. Specifically, we looked at onion cells, but this practical can be used to look at different types of both animal and plant cells, as well as to compare the differences between them. The key steps of this practical were slide preparation, microscope usage, and observation and drawing. Our key takeaways for future practical are to handle specimens and stains with care to avoid contamination or damage, use the microscope correctly to obtain clear and detailed images, and accurately draw and label cell structures as part of scientific documentation.
Watch the rest of the GCSE biology course at Sophos Education.